DNA lessons. In the first lesson, we learned about how DNA can be duplicated. The key to duplication is the pairing rules that we find on the rungs of the DNA ladder. A's and T's stick together, G's and C's stick together, so that when the two strands separate, the spare letters can find their matching partners and so make two identical molecules. Of course, that's critical for delivering hereditary information into daughter cells or from parents delivering hereditary information to their offspring. But now we get to the point, what is this information? How is it stored in the molecule? And what we'll see is the information is stored as this sequence of letters. So not the pairing of the rungs. The information content of the molecule consists of the sequence of letters along one strand. And that sequence of letters will be a code for how to build a protein. And so we call it a recipe in a sense. Now to us it looks like a random string of letters, but to the cell this means something. It will direct the cell's machinery to assemble the protein in a certain way. Now to figure out how that works, we need to take a look at what a protein is a little more carefully. Here we have a little model of a protein. It's a bunch of popcorn kernels on a string, and each kernel would represent one of the building blocks of proteins, and those are molecules called amino acids. So here we see an amino acid, and there are 20 different kinds of amino acids that make up the proteins in our cells. So a protein is a chain molecule, right? A chain of a different set of amino acids, and it might be a couple of amino acids long. Some proteins are hundreds of amino acids long, some are even over 1,000 amino acids long. When the cell makes a protein, it makes this chain of amino acids chemically bonded together, and then the whole thing folds up into some complex three-dimensional shape, and the shape then determines what its function is going to be. So if we have two amino acid sequences here with the different colors, green would be a different amino acid from a blue, a black, and red. This sequence of amino acids is different from this sequence. And that means that they will fold up differently, and when they fold up differently, then they will interact with other cell parts in a different way, so they'll have a different function. So the key to a protein's function is its shape, and its shape is largely determined by the amino acid sequence. So proteins with different sequences of amino acids will have different functions. Well, the very structure of a protein, being a sequence of amino acids, suggested a basic relationship then between the sequence of letters of DNA and proteins. And here the idea can be pictured here. So here's our nucleus, and here's a stretch of DNA. Notice the two strands have separated. The idea would be that the sequence of letters along one strand of DNA, maybe that was a code for the sequence of amino acids in the protein made from that recipe. Now remember, since DNA hangs around in the nucleus, we need to have a temporary copy of that information, and that is none other than this green little molecule, which is Hammerling's information molecule, right? So messenger RNA is going to be a temporary copy of the DNA sequence, and the idea is that sequence of letters will guide the cell into constructing the protein one amino acid at a time. So the sequence of amino acids is determined by the sequence of letters along messenger RNA, which is a copy of the sequence of letters in one strand of DNA. So that's the overall picture, that the key to information storage of the sequence of these letters is that proteins are a chain molecule, sequence of amino acids, and so we can have one sequence coding for another sequence. All right, so in summary, the sequence of DNA letters and its copy in messenger RNA specifies another sequence, the sequence of amino acids that make up proteins. Now, to decipher the language, we need to dig a little deeper. So in this diagram we see the three chain molecules for life on Earth. We have DNA up here at the top. Now we're only looking at one strand of DNA, so its complementary strand, right? Its strand that has its matching partners is not shown here. Then we have messenger RNA. Now we'll get into why it looks a little bit different than DNA in a moment. And then down here we have the amino acid sequence. Now these are abbreviations for the different amino acids. We have methionine, arginine, serine, glycine, serine, histidine, proline. We'll talk about stop in a second. The puzzle facing biologists was exactly how does this sequence specify the amino acids? Maybe one letter of DNA specifies or codes for the first amino acid. So the first letter in a gene, which would be like a recipe for a protein, maybe the first letter stands for the first amino acid in the protein. But of course, a little thought leads you to the conclusion that can't be, because there's only 4 different kinds of letters. There are T's, there are A's, there are C's, there are G's. But there are 20 different kinds of amino acids. So you wouldn't have enough words in your DNA language to specify all the 20 amino acids. So it can't be that a single DNA letter specifies the position of the amino acid. Well, what about a sequence of 2 letters? Well, when you do the math, you find out that there are a total of 16 possible 2-letter sequences for DNA. Again, that's not enough words, not enough sequences to specify 20 amino acids. And so researchers surmise that a plausible code, one that might work, would be sequences of 3 DNA letters would be sufficient to specify a particular amino acid. So the idea is the first 3 DNA letters codes for or stands for the identity of the first amino acid. The second 3 letters codes for or stands for the identity of the second amino acid. The third 3 letters, the third position in the protein. The next 3 letters, the fourth position in the protein. So again, and this is in fact the case, that we now know that it is triplets. So 3-letter sequences of DNA are the words in the language of biology. Each 3 letters specifies a certain one of the 20 amino acids that make up proteins. Okay, so now let's take a look at how this might actually be done in the nucleus, how a strand of DNA can be copied into a strand of messenger RNA, which then will go out into the cytoplasm to be used to build a sequence of amino acids, which is a protein. Here we see a diagram. Here's a gene. So a gene we're going to define as a sequence of DNA that has the recipe for a protein. Notice we've got this bit of DNA over here kind of wrapped up, so its information is not accessible at this moment. But here we have an accessible gene, and when a cell is going to make a protein, this protein, it has to unravel, unwind, and separate the DNA at that location. So that makes the information on one strand accessible. Now again, in the nucleus are spare parts. So just as we saw when DNA is duplicated, there are spare DNA letters. When a gene is going to be used to make a messenger RNA molecule, there are spare messenger RNA molecules around. Messenger RNA is composed of 4 letters, just like DNA, except there's one little difference. RNA has an A and a G and a C, but instead of a T, it has a U chemical called uracil. Now we'll just have to remember that, but otherwise the pairing rules are similar. So when there's a T in DNA, the corresponding RNA letter that sticks is an A. When there's an A, however, it doesn't have a T, it has a U. When there's a C in DNA, the RNA will be G, and when there's a G, there's a C. So what we see in this diagram then is a gene has been opened up, its information is now accessible, messenger RNA molecules being created using one strand of DNA as the sort of template. This would be a complementary strand of messenger RNA, and here we see the first 3 letters are beginning to detach. So this was a T, it matched up with the A. The A was right there, the U was right here next to the A, and here's the G, it was here with the C, but now it's starting to detach. And when this messenger RNA molecule is finished, it will detach from the DNA and leave the nucleus. Then the 2 DNA strands will get back together again to form the double helix, the twisted ladder. Here's another picture of that. Here we see the double-stranded DNA molecule. It has opened up at this region, so we'll call that a gene. Here is the information to build a protein, and it is now being used to make a messenger RNA copy of that information. Again, you see the pairing rules for RNA. If there's a DNA T, there's an RNA A. T, A, G, C, G, C, A, U. This red one is the messenger RNA. The two blue ones are the two strands of DNA, but notice only one of the strands of DNA is being used as a template to build the messenger RNA. Now remember from Hammerling's work, this process is happening inside the nucleus. These little messenger RNA molecules are Hammerling's information molecules. They leave the nucleus and come out to the cytoplasm. They're going to be used out in the cytoplasm here to direct the construction of the protein that they code for. So whenever a cell is making a protein, it has to first access the gene, access the information of that gene, make a copy of that information in messenger RNA, send that messenger RNA out into the cytoplasm, where it will hook up to the cell's machinery, and we saw that a little earlier. Here we see this structure is called a ribosome. You'll learn more about that in later grades. But here that structure is sort of like a protein factory, and it's going to read the RNA message. And that RNA message is going to instruct the factory to construct the protein one amino acid at a time. So the sequence of the amino acids is dictated by the sequence of the messenger RNA letters, which is dictated by the sequence of DNA letters. Okay. All right, so where were we? We are going to, so now this sequence of RNA letters, how do we know what the sequence of amino acids will be? Well, for that, we need to use the genetic code. Okay, this is the genetic code. This table tells us the relationship between the letters, the sequence of letters in DNA and RNA, and the amino acids that make up proteins. Now, actually, this particular display is in the language of RNA because we see the U. But you'll notice around the ring here, we have all the 20 different amino acids. These are the names of the amino acids that make up proteins, the building blocks of proteins. So here's how it would work. Let's say we have a sequence of RNA letters here. Now, this would be a copy of the one strand of DNA, but that's not shown here. Here we just have the RNA sequence. We have 3 letters. Remember, 3 letters of DNA specifies an amino acid. So too with RNA. 3 letters of RNA, A, U, G. So these 3 letters will specify the first amino acid at the first position in the protein. So let's see how we would identify that. So it's A, U, G is the sequence of letters. Well, now we come to our diagram. We start in the center, the large letters. This center region corresponds to position number 1. A, since the triplet was AUG, the first position is an A, we go to the A quadrant. So now we're in the A quadrant. We don't now care about all the rest of this. We're in the A quadrant. The second position was U. So within the A quadrant, we find the second position of U. Here it is here. So the second ring is the second position. And the third letter was G. So we come out to the outer ring. So AUG. That indicates that methionine would be the first amino acid. Now, if the third letter was AUA, then isoleucine. If the third letter was C, it would also be isoleucine. And if it was U, it would also be isoleucine. So notice there are 3 triplets here that correspond to isoleucine. And that's very common as you look around the code here. Let's take a triplet CCU. That would code for proline. But so also would CCC, CCA, CCG. So there's redundancy in the genetic code. And we can think about that mathematically. Remember, since we're dealing with triplets here, if you ask yourself how many possible 3-letter sequences are there? Well, for DNA having 4 letters, there are 4 different letters that could go in the first position, 4 different letters that could go in the second position, 4 different letters that could go in the third position, 4 times 4 times 4, 64. There are 64 possible 3-letter sequences. Yet there are only 20 amino acids. So there's going to be redundancy. We have 64 words in the language of biology to specify or code for 20 different amino acids. And that's why you have multiple triplets standing for or coding for each amino acid. So we could go to triplet number 2, UAC. We come back to our code. Now we start at the U. UAC is going to be tyrosine. If it were UAU, it would also be tyrosine. GAG, we go to G, then the A, and the G, glutamic acid. and so on. So you just go down one triplet at a time. Let's take a look at this last one, UGA. UGA. Notice that's something different. That says stop. Now that's not an amino acid. It's actually sort of punctuation. It means that the message is finished. So each messenger RNA will have a stop triplet at the end, and that tells the cell that the recipe is finished, and then the protein will detach from the factory, and then it will fold up. So there's a start to the message. The first three letters of the messenger RNA specify the first amino acid, and then the last three letters of the messenger RNA specify the last amino acid in the protein. Now, Darwin would be very excited to learn about this because all living things on Earth utilize the same genetic code. Again, the code refers to this relationship between DNA letters and the amino acids in proteins. Of course, this is the language of RNA here, but the general idea is that relationship between genetic information and the structure of proteins. This same genetic code is shared by all living things. And Darwin would explain that by saying that we have inherited this language from our common ancestor. The earliest living things evolved this code, and all species that later evolved have inherited this code.